Under 3 feet of solid Arctic ice, in total darkness, at temperatures cold enough to freeze your blood, something is alive. And it is doing something that biology said was impossible here. Scientists just found bacteria producing fertilizer in the frozen Arctic Ocean, with 0 sunlight. These tiny organisms are changing how carbon moves through our entire atmosphere. We will go from the moment the discovery broke to what it means for your future on this planet. If you enjoy this journey, subscribe and hit like. It keeps us going. Stay with us. We begin. The email arrived on a Tuesday morning in October 2025. A research team at the University of Copenhagen opened their lab results. They stared at the numbers. Then they stared again. The data showed something the Arctic Ocean was never supposed to do. Deep under the frozen surface of the world's most remote sea, something tiny was converting raw nitrogen gas into fertilizer. Biological fertilizer. The kind that feeds entire food chains. The kind that, according to every textbook ever written, required warm water and sunlight to produce. The Arctic had neither. The water temperature at the sampling sites hovered at 28 degrees Fahrenheit. That is below freezing. The ice above the samples stretched 3 feet thick in solid sheets. 0 sunlight reached the water below. Scientists had spent decades assuming this environment was biologically dead, at least for this particular chemical process. They were wrong. Lead researcher Lisa von Friesen later said that science had completely misread the limits of these organisms. Her team had not gone to the Arctic expecting this. They had gone to map what was already known, to fill in the edges of existing models. Instead, they found a process operating in plain sight, hidden under ice, in the dark, for who knows how long. Here is why that moment matters so much. Nitrogen fixation is one of the most important biological processes on Earth. Every plant, every algae, every creature at the base of every food chain depends on it. Nitrogen is everywhere in the air and the water, but living things cannot use it in its raw form. Something has to convert it first. That something is usually a specific type of bacteria, and those bacteria were thought to need warmth and light to function. The Arctic provided neither. So for decades, scientists wrote the region off. Imagine building a map of a country, but leaving an area the size of the entire United States blank because you assumed nothing was there. Then someone walks into that blank space and finds a city, an entire functioning city, running full operations, completely off the grid. That is roughly what happened here. The team had deployed 2 massive icebreaker ships to collect samples across 600 miles of frozen ocean. The ships were named the IB Odin and the RV Polarstern. They are among the most powerful research vessels ever built, capable of crushing through ice that would stop most steel ships cold. Even with that equipment, reaching the sampling sites required weeks of navigation through some of the most dangerous water on the planet. 13 locations, 600 miles apart. Every single one showed the same result: active nitrogen fixation. Confirmed. The bacteria were not dormant. They were working, producing, cycling nutrients through the water in a loop that fed the algae above them and received waste from that same algae in return. A hidden engine, running continuously in a place science had labeled empty. And the consequences of that engine running, undetected for all this time, those reach far beyond the Arctic, far beyond the ocean. They touch the air above your head right now. But before we get there, you need to understand exactly why biology said this was impossible, and why an entire scientific history nitrogen fixation came with 2 hard rules. Rule 1, you need warmth. The enzymes inside nitrogen-fixing bacteria are fragile. Cold temperatures slow them down. Extreme cold, the kind that drops water below freezing, shuts them off entirely. At least, that was the assumption every major biology course repeated for decades. Rule 2, you need energy. Most nitrogen-fixing bacteria use sunlight to power the chemical reaction. They are a type called cyanobacteria, and they run on photosynthesis the same way plants do. Take away the light, and the reaction stops. The Arctic ice blocks almost all sunlight. What little reaches the water is dim, scattered, and seasonal. For months each winter, there is no light at all. Put those two rules together, and the conclusion writes itself. The deep Arctic Ocean, cold and dark year-round, should produce zero nitrogen fixation. Scientists accepted this conclusion and moved on. Research funding flowed to the tropics. The Arctic was assigned a number close to zero and left alone. That assumption sat unchallenged for decades because nobody seriously looked. Here is the problem with assuming an environment is dead. You stop collecting evidence, and when you stop collecting evidence, absence of data looks identical to absence of life. The Arctic did not prove the textbooks right. The textbooks simply never went back to check. Their samples came from the water column directly beneath the sea ice, pulled up through the frozen surface, at 13 sites spread across the central Arctic and the waters north of Svalbard and northeast Greenland. The equipment they used could detect nitrogen fixation at extremely low concentrations. Previous Arctic expeditions had used less sensitive tools that would have missed exactly what this team found. The bacteria they discovered belonged to a group called non-cyanobacterial diazotrophs. Long name. Simple concept. They are nitrogen fixers that run without sunlight. Instead of photosynthesis, they use dissolved organic matter floating in the water around them. Dead algae cells, microscopic waste products, chemical scraps that drift down from the ice above. They eat the leftovers. Then they run the chemistry. The two textbook rules still hold for the bacteria scientists knew about. Warm water, sunlight. Those rules apply perfectly to cyanobacteria in tropical and temperate oceans. The mistake was assuming those were the only bacteria doing the job. Beneath the Arctic ice, a completely different group had evolved a completely different strategy. Same end result: nitrogen gas converted into ammonium. Different fuel source, different temperature range, different environment entirely. Every climate model that ignored Arctic nitrogen fixation now carries a gap. Every ocean simulation that set polar nitrogen input to near zero is running on flawed data. The researchers calculated that the highest rates of bacterial activity in their samples reached up to 5 nanomoles of nitrogen per liter of water per day near the ice edges. That sounds small. Multiply it across the surface area of the entire Arctic Ocean, and it stops sounding small. Scientists now face the task of rebuilding their models from scratch. And the bacteria doing all of this, their genetic code looks nothing like what researchers expected to find. What is inside those cells is strange enough to suggest something far older and more resilient than anyone guessed. Picture a farmer in January, frozen ground, no sun. He walks out anyway and spreads a bag of fertilizer across his field. Come spring, that field explodes with crops. That is nitrogen fixation. Except the farmer is a bacterium the size of 1/1000th of a grain of sand. The field is the Arctic Ocean, and the fertilizer is a molecule called ammonium. Here is how it works. The air above any ocean is about 78% nitrogen gas. The water below absorbs some of that gas. Nitrogen is everywhere, but in its gas form, living things cannot use it. A fish cannot absorb nitrogen gas and grow from it. Algae cannot either. The nitrogen is present, but locked up, useless to almost every living thing on the planet. Certain bacteria carry a special set of genes. Those genes produce a protein called nitrogenase. Nitrogenase grabs nitrogen gas molecules and breaks them apart. Then it attaches hydrogen atoms to the nitrogen, creating ammonium. Ammonium dissolves easily in water. Algae absorb it directly. Ammonium is, for marine life, one of the most valuable nutrients that exists. Without it, ocean food chains collapse at the base. Every large fish you can think of, Every whale, every seal, every seabird that dives for prey traces its energy back down through the food chain to algae. Algae need ammonium to grow. Ammonium comes from nitrogen-fixing bacteria. Remove the bacteria, and the entire pyramid above them starves. In tropical oceans, this process runs constantly. Warm water, abundant sunlight, cyanobacteria thrive. The food web stays fed. Scientists understood the system well and modeled it carefully. The Arctic looked different. Cold temperatures, no light under the ice. So researchers assumed the nitrogen pipeline shut down at the poles. Arctic food chains, they thought, ran on nutrients recycled from old organic matter and whatever currents pushed up from deeper, warmer water. The bacteria the Copenhagen team found proved that assumption was incomplete. These microbes run the same nitrogenase chemistry but power it differently. Instead of light, they consume dissolved organic carbon. Think of dissolved organic carbon as a kind of invisible soup floating in the seawater. It comes from algae growing on the underside of the ice, from dead cells breaking down, from microscopic waste products drifting through the water column. The bacteria absorb this soup, extract energy from it, and use that energy to run the nitrogen fixation reaction. The output is the same. Ammonium released into the water, the algae above them absorb that ammonium and grow. As the algae grow, they shed more waste, more dead cells, more dissolved organic material. That material sinks and feeds the bacteria again. The loop closes on itself perfectly, a self-contained factory running on its own waste, producing fertilizer in the dark, at temperatures that should make the whole operation impossible. The nitrogenase enzyme is one of the oldest biological machines known to science. It evolved billions of years ago, long before Earth had the oxygen levels it carries today. Finding it running under Arctic ice suggests these bacteria may have been doing this for far longer than anyone imagined. And how long they have been doing it matters enormously, because every year they ran undetected, scientists built their climate models without them. The size of that error is only starting to come into focus. Science follows money. Money follows assumptions. For most of the 20th century, the assumption was simple. Warm oceans are active. Tropical and subtropical waters received research ships, funding grants, and decades of detailed study. The nitrogen fixation literature was built almost entirely on data from the Atlantic Ocean, between the tropics and from the Pacific near Hawaii. The Arctic received almost nothing by comparison. Part of the reason was practical. Reaching the central Arctic Ocean requires icebreaker ships. Those ships cost enormous amounts of money to operate. A single research expedition to the high Arctic can cost as much as running a university biology department for several years. Tropical expeditions are cheaper, safer, and logistically far simpler. Results flow faster. Papers get published. Funding cycles reward that speed. The other reason was intellectual. Scientists believed the cold, dark, nutrient-poor conditions under Arctic ice were incompatible with the energy demands of nitrogen fixation. So even when small amounts of funding became available for polar research, nitrogen fixation was rarely the priority. The assumption made the question seem already answered. This created a feedback loop. No data came in from the Arctic. Models confirmed that the Arctic contributed nothing meaningful to global nitrogen. Those models shaped the next generation of researchers who designed their studies around the tropics again. The blind spot grew. Early hints appeared around 2017. Some marine chemistry studies in polar waters showed nitrogen levels that seemed slightly higher than the recycled nutrient model predicted. A few researchers noted the gap but lacked the tools and the funding to pursue it. The sensitive equipment needed to detect low-level nitrogen fixation was expensive and new, and the central Arctic, covered in multi-year ice, was difficult and dangerous to reach. The Copenhagen team represented a shift. They had newer detection tools. They had access to two of the most powerful icebreakers in the world. And they had a specific scientific question. Was the Arctic truly contributing zero nitrogen, or was something else going on? They sailed into the central Arctic Ocean in 2023 with that question. The IB Odin and the RV Polarstern covered over 600 miles of frozen ocean. They sampled at 13 distinct sites. They analyzed water from below the ice, from the edge zones, from deep into the central basin. And the answer came back. Something else was definitely going on. The decades of tropical focus had produced genuinely important science. Understanding how nitrogen fixation works in warm oceans gave researchers a strong foundation, but it also created an invisible wall around the model. Anything that did not fit the tropical template got filtered out. A process running in the Arctic, without light, without warmth, simply did not compute. The cost of that blind spot is now measurable. Every climate prediction made over the last 30 years, that included ocean carbon absorption, was built on incomplete data. How incomplete? Scientists are still calculating. The total volume of nitrogen these Arctic bacteria produce each year across the entire ocean has never been measured. That number is still unknown, and the bacteria doing this, the ones at the center of the whole discovery, carry a genetic signature unlike anything researchers expected to find in polar water. When the Copenhagen team pulled their water samples onto the ship and processed them for genetic material, they expected ambiguity. Polar environments often contain DNA from dozens of sources. Cells drift. Currents carry genetic material from thousands of miles away. Finding active nitrogen fixes in the sample would have been remarkable enough. What they found was far stranger. The genetic signatures of the bacteria matched a group called non-cyanobacterial diazotrophs. That phrase breaks down simply. Non-cyanobacterial means they are not the sunlight-powered nitrogen fixers scientists already knew. Diazotroph means nitrogen fixer, so the label translates to a nitrogen-fixing bacterium that runs without photosynthesis. Scientists knew this group existed. They had found them in temperate and even some deep ocean environments before. But the Arctic, in water below freezing, that was a different category entirely. The key genes researchers looked for are called NIF genes. These genes carry the instructions for building nitrogenase, the protein that actually breaks nitrogenase apart and converts it to ammonium. In the Arctic samples, the NIF genes were present and fully active. The bacteria were expressing those genes at full capacity, producing working nitrogenase enzymes, and running the conversion process continuously. This was confirmed, not a theory. The genetic machinery was intact and operating. What made the genetic picture even stranger was the divergence from known tropical relatives. The Arctic bacteria shared the core nitrogen-fixing genes with their warm water cousins, but showed significant differences across the rest of their genome. Their cold adaptation machinery looked like a separate evolutionary path. The genes controlling membrane flexibility, enzyme stability at low temperatures, and energy management under dark conditions all showed modifications that tropical versions of the same bacteria do not carry. Bacteria evolve fast. A single bacterial generation can take as little as 20 minutes under ideal conditions, but evolving a complete cold-dark adaptation package takes enormous amounts of time and pressure. The genetic distance the Copenhagen researchers observed suggests these bacteria did not drift into the Arctic recently and survive by accident. They adapted over a very long period, perhaps millions of years. The implications of that timeline are significant. If these bacteria have been living and working under Arctic ice for millions of years, then the nitrogen input they provide to the polar ocean is ancient. It predates modern climate models by a geological timescale. It predates the research ships. It predates human civilization. The Arctic Ocean has been receiving this invisible fertilizer for longer than our species has existed, and science had no idea. The gene sequencing also confirmed that the bacteria were locally adapted, meaning they were permanent residents of the Arctic environment. not temporary visitors blown in by ocean currents. They reproduced there. They thrived there. The community under the ice was self-sustaining. One detail from the genetic analysis stood out to researchers above all others. The specific enzyme variants these bacteria used to run nitrogen fixation at sub-zero temperatures appear to be unique to polar environments. The same basic nitrogenase design, but with molecular modifications that allow it to stay flexible and active when everything around it is close to freezing. Nobody knew those modifications existed before this study. And the way these bacteria survive, the actual energy deal they make with the ocean around them, turns out to be one of the most elegant biological arrangements ever discovered under ice. Every living thing leaves waste behind. Algae growing on the underside of Arctic ice shed dead cells, release dissolved sugars, and produce microscopic organic particles that drift down into the water below. In a warmer ocean, this material gets consumed quickly by a wide range of organisms. In the freezing dark of the Arctic under ice zone, the list of things that can eat it is much shorter. The non-cyanobacterial diazotrophs eat it. These bacteria are heterotrophs, meaning they get their energy from consuming organic compounds rather than from sunlight. The dissolved organic carbon floating through the cold Arctic water column is their fuel source. They absorb it, break it down through internal chemistry, and extract enough energy from that process to power their nitrogen-fixing machinery. The nitrogen-fixing reaction is expensive. It takes more biological energy per molecule than almost any other chemical process a bacterium performs. In tropical cyanobacteria, sunlight provides that energy in abundance. In the Arctic, these bacteria squeeze that energy out of dead organic matter instead. Picture a car engine running on cooking oil instead of gasoline. Same basic output, completely different fuel source. The engine works, just through a different set of reactions. The bacteria process the organic waste and use the energy to convert dissolved nitrogen gas into ammonium. They release that ammonium directly into the surrounding water. The ammonium drifts upward through the water column and reaches the algae growing on the ice above. The algae absorb the ammonium instantly. Ammonium is to algae what fertilizer is to a crop field. It accelerates growth. More algae grow on the ice surface and along the water column. As those algae grow, reproduce, and eventually die, they shed more organic material back into the water below. That material sinks. The bacteria eat it. They convert more nitrogen. More ammonium rises. More algae grow. The loop runs itself. What makes this system remarkable is its efficiency. The waste product of one organism is the energy source of another, and the waste product of that second organism is the essential nutrient of the first. Nothing escapes the cycle. Everything gets used. The whole operation runs continuously in a place where most biology is expected to struggle just to survive. The scientific term for this kind of arrangement is a mutualistic loop. Both species benefit directly from each other's presence. Remove either one, and the whole system degrades. The algae without bacteria would eventually exhaust the available ammonium and grow more slowly. The bacteria without algae would lose their carbon fuel source and slow their nitrogen fixation. Neither organism planned this. It emerged over millions of years of shared evolution in the same extreme environment. The sampling data confirmed that the highest rates of bacterial activity appeared in the water directly beneath the densest concentrations of ice algae. The relationship was not random or coincidental. The bacteria clustered where the food was richest, and the food was richest where the algae were most active. This invisible partnership powers something much larger than itself. The ammonium it generates does not stay in the under-ice zone. It mixes into the broader water column and feeds organisms across the Arctic ecosystem. And the place where this partnership runs hottest, the most chemically active zone in all of the Arctic Ocean, is somewhere most scientists would have assumed was too dangerous and unstable to support steady biological work. Under the Arctic ice, two life forms run a deal that neither one chose. The algae live on the underside of the ice and just below the surface of the water. They are single-celled. They photosynthesize using whatever dim light filters through the ice in spring and summer. They grow in thin brownish-green mats that coat the frozen underside of the ocean's ceiling like moss on the underside of a rock. The bacteria live in the water below them. No light reaches that far. The bacteria do not photosynthesize at all. Together, they run one of the most productive biological exchanges in the polar world. The algae grow and eventually shed cells. Old cells break apart. Sugars and proteins and dissolved organic molecules drift downward into the dark water. The bacteria are waiting. They absorb this material and run their nitrogen-fixing chemistry on the energy extracted from it. The product of that chemistry is ammonium, which rises back up into the water column where the algae can absorb it. The algae take in the ammonium and grow faster. More growth means more organic material shed downward. More material means more energy for the bacteria. More bacterial energy means more ammonium released upward. Each organism amplifies the other. Researchers confirmed this relationship through the sampling data. The 13 sites stretched across 600 miles of the central Arctic Ocean, from the waters north of Svalbard to the outer zones northeast of Greenland. At every location, bacterial activity tracked closely with algae density. Where algae concentration rates were also highest, the two measurements moved together like two instruments in the same song. The relationship also holds up across seasons. Arctic algae have a strong seasonal pulse, growing intensely during the months when some light reaches them through the ice, then slowing through the polar winter. Bacterial nitrogen fixation in the samples tracked that pulse, accelerating when organic material from algae growth was abundant and slowing when the algae retreated. This seasonal synchrony suggests the two communities have been coordinated for a very long time. They have adapted to each other's rhythms across millions of years in the same environment. What happens at the edges of the ice matters even more. Along the boundary where solid ice meets open water, conditions are more extreme and more dynamic. Ice melts and refreezes. Algae bloom intensely in the meltwater. Organic material floods the edge zone. Bacterial nitrogen fixation reaches its peak rates right along this boundary, confirmed by the sampling data. The ice edge is the hottest engine in the whole system, and that engine is getting larger. As global temperatures rise, the Arctic ice retreats every summer. The boundary between solid ice and open water stretches across a longer and longer line each year. The zone where bacterial activity peaks is physically expanding. More melt edge means more habitat for peak nitrogen fixation. The chemistry of the Arctic Ocean is shifting in real time. The algae and bacteria partnership, operating invisibly for perhaps millions of years, is now scaling up. Its output is increasing. The ammonium it produces is feeding a growing bloom of algae across the Arctic. And that growing bloom is doing something to carbon dioxide in the ocean above it that will matter to every person on the planet, whether they ever see the Arctic or not. The IB Odin weighs over 13,000 tons. Its hull is reinforced steel nearly 2 inches thick. Its engines produce enough power to crush through ice 3 feet thick at a steady walking pace. Standing on the deck as it moves through pack ice, you hear a constant grinding roar beneath your feet as the ship splits frozen ocean apart and pushes the broken pieces to each side. This is the machine that took the Copenhagen research team to the Central Arctic. The RV Polarstern accompanied the Odin on the expedition. The Polarstern is a German research vessel with over 40 years of polar expedition history. Its laboratory spaces are built to maintain precise temperature controls even while the ship pitches through rough seas. The ship can carry dozens of scientists and operate continuously for months without resupply. Together, these two ships formed the platform for the 2023 expedition that produced the groundbreaking data. Inside an icebreaker research vessel, the environment is controlled chaos. The labs run around the clock. Scientists work in 8-hour shifts, because the sampling windows do not stop for sleep schedules. The ship holds position at a site, and researchers have a fixed window to lower equipment through holes in the ice or through the water directly off the hull. The cold is relentless, even inside the ship. Working near open doors or on deck during sampling operations means exposure to temperatures well below 0 Fahrenheit. Every sample collection requires protective gear. Equipment that would function normally at room temperature can malfunction in minutes when exposed to Arctic air. The sampling equipment used for nitrogen fixation measurements is sensitive and delicate. Researchers lower sealed collection tubes on long cables through holes drilled in the ice or directly off the ship's stern. The tubes must reach specific depths in the water column without contamination. Any contact with the ship's own chemical wake can corrupt the results. Positioning the tubes cleanly requires precision and patience. The collected water samples go immediately into the ship's cold laboratory, where they are processed at temperatures close to what the Arctic water itself reaches, to prevent the bacteria inside from behaving differently than they would in their natural environment. Genetic material is extracted on board. Nitrogen fixation rates are measured using radioactive tracer methods that track exactly how much nitrogen gas the bacteria convert over a defined time period. The data from 13 different sites across 600 miles of ocean had to be collected, processed and stored with consistent methodology across weeks of travel through changing ice conditions. What the team confirmed across all 13 sites, despite the distance between them and the varying ice conditions at each one, was a consistent pattern: active nitrogen fixation every time. The consistency of the signal across that geographic spread was itself a major finding. A single anomalous site could be explained away. 13 sites across 600 miles of Arctic Ocean cannot be. The geographic range of the sampling tells a different story than the bacteria alone. It says the process is not a local quirk in one unusual spot. It is a feature of the entire central Arctic, and the most extreme readings in the dataset came from a place where the scientists nearly expected the biology to stop entirely. The process sounds simple. It is anything but. A research team drills a hole through 3 feet of solid Arctic ice. The drill bit screams through frozen ocean as ice chips spray across the white surface. The hole opens into darkness below. The water sits at 28 degrees Fahrenheit, 2 degrees below the freezing point of fresh water. The saltier polar seawater stays liquid at that temperature, but just barely. Researchers then lower a series of precisely sealed collection bottles on a cable through that hole. Each bottle is designed to stay closed during descent and then open at a specific depth to collect a water sample. The cable has stops attached at measured intervals, so each bottle captures water from a different level of the water column. One bottle might collect from just below the ice, another from 30 feet down, another from 60. The depth profile matters because bacterial activity varies through the column, and mapping that variation is part of the study. After a set time, the cable comes back up. Researchers pull the bottles onto the ice surface and move them immediately into insulated containers. Any warming of the sample, even by a few degrees, can change the behavior of the microbes inside and corrupt the nitrogen fixation measurements. Inside the ship laboratory, the water from each bottle goes through a series of analyses. One of the key methods used is called the acetylene reduction assay. This technique introduces acetylene gas into the sample and tracks how quickly the bacteria convert it into a measurable byproduct. Since the same enzymes that fix nitrogen also convert acetylene in a predictable way, the rate of conversion reveals how actively the bacteria are fixing nitrogen in real conditions. It is a way of eavesdropping on the bacteria's chemistry without disrupting it. The team also used stable nitrogen isotope tracing. Researchers introduced a labeled version of nitrogen gas into sealed samples and then measured, over time, how much of that labeled nitrogen appeared as ammonium. This method directly confirmed that the bacteria were doing the conversion actively, producing real ammonium rather than just carrying the genes for it without expressing them. Both methods confirmed the same result. Confirmed. The sampling campaign covered 13 distinct sites across the Central Arctic Ocean and the Eurasian Arctic Basin. Some sites sat in multi-year ice, frozen so long that it had compressed into dense blue-tinted sheets. Others sat at the ice edge, where active melting created a dynamic mixing zone. The variety of conditions let researchers understand not just whether nitrogen fixation was happening, but where it was happening fastest, and why. Near the ice edges, rates reached up to 5 nanomoles of nitrogen per litre per day. In the central pack ice regions, rates measured between 0.4 and 2.5 nanomoles per litre per day. Those numbers are small, but they apply to a water column, not a single cup. Multiply them through the full depth of the water sampled, then across the millions of square miles of the Arctic Ocean, and they describe a massive annual nitrogen input that current global models do not account for. The scientists aboard those ships had just measured a gap in humanity's understanding of how the planet works. And back on land, the climate modeling community was about to confront exactly how large that gap was. 600 miles of frozen ocean, 13 stops, each one a different window into the same hidden system. The expedition mapped the Central Arctic Ocean and the Eurasian Arctic Basin, covering the waters north of Svalbard and stretching northeast toward Greenland. This region sits at the top of the planet, locked under ice for most of the year and accessible to large ships only during a narrow summer window. The sites were chosen to capture the widest possible range of ice and water conditions. Researchers wanted to know if nitrogen fixation was a local event tied to one specific set of conditions, or if it was a broad regional feature. The answer required sampling across geography, ice thickness, water depth, and proximity to the ice edge. Some sites sat in the heart of the pack ice. This is the oldest, thickest ice in the Arctic, compressed and hardened over multiple winters into dense floating sheets. The water beneath it receives almost no light at any time of year. The bacterial activity here ran at lower rates, between 0.4 and 2.5 nanomoles of nitrogen per litre per day, but it ran consistently. The cold and dark environment that was supposed to shut down this chemistry entirely was not shutting it down. Other sites sat near the ice edge, where the boundary between frozen and open water shifts with wind and temperature. These locations were more dynamic. Ice would melt back during warm periods and reform during cold ones. Algae bloomed intensely in the meltwater. Organic material was abundant. Bacterial activity at these sites reached the expedition's highest measured rates, up to 5 nanomoles per litre per day. The contrast between the pack ice sites and the edge sites told researchers something important. The bacteria adapt their activity level to the resources available. Where more organic material flows from algae, bacterial nitrogen fixation runs harder. Where resources are scarce, the bacteria still run, but more slowly. The process is responsive to conditions rather than locked at a fixed rate. The depth profile also varied between sites. At some locations, the highest bacterial activity concentrated just below the ice, within the first few feet of the water column, close to where algae grew most densely. At others, activity spread through a deeper water column, suggesting that organic material was sinking further before being consumed. The geographic spread of active sites, from near Svalbard to the waters northeast of Greenland, covered a stretch of ocean roughly equal to the distance from New York City to Chicago, then on to Denver. Every single stop on that route showed positive nitrogen fixation. That geographic consistency destroyed any argument that the results represented an isolated anomaly. The process was regional, the bacteria were everywhere under the ice. What the dataset could not yet answer was the total. 13 sites give researchers a strong signal. They do not give the total annual nitrogen contribution of the entire Arctic Ocean. Extrapolating from 13 points to an ocean that covers 5.5 million square miles requires modeling assumptions that scientists are still developing. That total remains one of the most important unknown numbers in current climate science. But even without the total, the direction of the finding was clear, and the most dramatic evidence came from the sites where conditions were most extreme, the places where ice met open water, and the whole system ran at full speed. Draw a line across the Arctic Ocean. On one side, white, solid ice, a frozen ceiling over dark water. On the other side, blue, open ocean, sunlight hitting the surface directly, wind creating waves. The line between them is called the ice edge, and it is, according to the data from the Copenhagen expedition, the most biologically active zone in the entire Arctic Ocean for nitrogen fixation. The reason comes down to resources. When ice melts along its edge, it releases fresh water into the ocean. That fresh water is slightly less dense than the salt water below it, so it floats near the surface. This creates a thin layer of stable, calm water right at the top of the ocean near the ice boundary. Sunlight hits that layer directly. Algae bloom explosively in it, sometimes turning the water a vivid green in satellite images taken from above. As the algae bloom, they produce enormous amounts of organic material, dead cells, shed proteins, dissolved sugars. This material sinks from the bright surface layer into the darker water below, and in that darker water, the nitrogen-fixing bacteria are waiting. The material that sinks from an ice edge bloom is far richer than what trickles down from algae growing on the underside of thick pack ice. The edge bloom is intense and dense, producing more organic material per square mile than the pack ice algae communities produce. The bacteria respond by running their nitrogen fixation at maximum rate. The sampling data confirmed this pattern precisely. Sites near the ice edge consistently recorded the highest nitrogen fixation rates in the study. The peak measurement of 5 nanomoles of nitrogen per litre per day came from water collected directly adjacent to the active melting zone. The ice edge is also dynamic in a way that the pack ice is not. Wind and temperature shift the edge position by miles in a single day. This constant movement creates a rolling wave of fresh algae blooms, each one feeding the bacterial communities below as it passes. The edge is never still. The biology runs continuously. Here is the part that connects to something much bigger. The Arctic ice edge is retreating. Every year, summer brings warmer temperatures, and the edge pulls further back from where it sat the year before. The minimum ice extent at the end of Arctic summer has been shrinking consistently for decades, confirmed by satellite measurements. The area of open water in the Arctic is growing. That means the length of the ice edge is also growing. A retreating edge does not simply disappear. As ice pulls back from one area, it creates a longer perimeter of active melting boundary. More edge means more algae bloom zone. More bloom zone means more organic material sinking into the water below. More organic material means more fuel for the nitrogen-fixing bacteria. The highest activity zone in the Arctic is expanding alongside climate change. Scientists had not included this feedback in their models. A warming Arctic was expected to affect the ocean in specific ways: more open water, changed circulation patterns, shifting fish populations. The bacteria accelerating nitrogen production along the expanding ice edge were an unaccounted variable, and the product of all that accelerating nitrogen fixation, the ammonium flooding into the water column, sets off a chain reaction that does not stay in the Arctic. It reaches upward through the food web and outward across the planet. The Arctic ice is retreating. That is confirmed by decades of satellite data, ship records, and direct measurement. Most people understand this as a loss. Ice disappears, habitat shrinks, polar bears lose hunting ground, sea levels feel the ripple effects. The news frames Arctic melting as a story of subtraction. The story of the nitrogen-fixing bacteria flips part of that frame. For them, the melting is expansion. The bacteria live in the water directly beneath the ice and along the ice edge zone. As the ice retreats, the active ice edge, the most productive zone for their food supply, stretches across a larger boundary. The area of water that sits adjacent to melting ice increases as total ice coverage decreases. The bacteria's most favorable habitat, the edge zone where organic material from algae blooms is richest, grows larger every summer. This is a paradox that climate scientists did not anticipate. A shrinking Arctic ice cap creates a longer perimeter. Think of it like a receding shoreline on a lake with an irregular coastline. As the water level drops, the exposed shoreline can actually grow longer even as the lake shrinks, because more curves and inlets open up. The Arctic ice edge behaves similarly as it retreats into more fragmented patterns. The 2023 expedition captured this effect in the sampling data. Sites near active melt zones consistently showed higher bacterial activity than sites in stable, thick pack ice. The more dynamic the ice condition at a site, the higher the nitrogen fixation rate. This dynamic has an important time component. Arctic sea ice reaches its minimum extent in September, at the end of the summer melt season. In the 1980s, the September minimum covered roughly 3 million square miles of ocean. By the early 2020s, that number had fallen to roughly 1.5 million square miles. The retreat has been roughly linear across that period, with some year-to-year variation. As that minimum shrank, the summer ice edge zone expanded, the algae bloom season in the meltwater lengthened, the window during which organic material floods the water column below the edge grew wider, the bacteria's best feeding season got longer. Researchers confirmed that the highest nitrogen fixation rates appeared at sites where the ice had been actively retreating during the expedition period. The biological response to melt is immediate. Within days of a melt event, bacterial activity in the water below can spike as fresh organic material arrives from the intensifying algae bloom above. The scale of this expansion matters when you try to calculate total nitrogen input to the Arctic Ocean. Scientists currently have measurements from 13 sites across 600 miles. Projecting those measurements across the full Arctic Ocean requires understanding how bacterial activity changes across all ice conditions. The retreating ice data provides part of that picture, but the total calculation remains unfinished. What is already clear is the direction. More melting leads to more bacterial activity. The Arctic nitrogen system is not a fixed background process. It is a responsive one, and it is responding to climate change by increasing its output. That increasing output does something specific to the algae population of the Arctic Ocean, something that drives a consequence visible from space. A molecule of ammonium is small enough that 1 billion of them could fit on the period at the end of this sentence. One molecule alone does nothing meaningful. But release enough of them into the water column of the Arctic Ocean, and you trigger a response that scales from the microscopic to the enormous. Ammonium is nitrogen in a form that algae can absorb directly through their cell walls. Algae are the base of the ocean food chain. They are the grass of the sea. Every animal above them, from tiny shrimp to massive bowhead whales, depends on algae either directly or through the chain of predators that eat them. In a nutrient-poor ocean, algae grow slowly. Limited nutrients mean limited cell division. The population stays small, the food chain above stays thin. When ammonium enters the water, the math changes immediately. Algae absorb the ammonium and use the nitrogen it carries to build proteins and DNA. Proteins and DNA are the machinery of cell division. More ammonium means faster cell division. Faster cell division means a rapidly growing algae population. The population grows until something else limits it, usually light or phosphorus, but in the nutrient-limited Arctic, ammonium is often the limiting factor. Remove the limitation and the population blooms. A bloom is an exponential event. 1 cell becomes 2, 2 become 4, 4 becomes 16. Within days, a patch of ocean that was relatively clear can turn deep green as millions of algae cells pack into every liter of water. These blooms are large enough to be visible in satellite imagery taken from hundreds of miles above the surface. The Arctic has always had algae blooms, particularly in spring, and early summer when light returns. But those blooms were constrained by available nitrogen. The discovery that bacteria under the ice have been supplementing the nitrogen supply suggests the blooms were larger than they would have been without the bacteria's contribution, and that they may grow larger still as the bacterial population expands along the retreating ice edge. A larger bloom feeds more zooplankton. Zooplankton are the tiny animals that graze on algae the way cattle graze on grass. More zooplankton feed more small fish. More small fish feed larger fish, seabirds and marine mammals. The chain runs all the way up to bowhead whales, which are among the largest animals on Earth and live year-round in Arctic waters. What begins with a bacterium producing a molecule of ammonium in total darkness ultimately moves through every level of the food web above it. The chain reaction takes weeks to fully propagate from the microscopic to the visible. A bloom that starts from bacterial ammonium in March may be feeding young fish by June and contributing to whale feeding behavior by August. The timing and size of that cascade affects commercial fishing stocks, indigenous community food supplies, and ecosystem health across the entire Arctic region. Scientists modeling the Arctic food web did not have the bacterial nitrogen input in their equations. Every prediction about how the Arctic food chain responds to climate change was built without this variable included. And what happens to the algae after they bloom has an effect that extends far beyond the Arctic Ocean itself. Spring arrives in the Arctic with weeks of accelerating light. The sun returns after months of polar winter, and the first photons to reach the ocean's surface hit algae that have been waiting. The algae respond immediately. They begin dividing. The bloom builds slowly at first, then faster, then in some areas explosively, as more light and more nutrients push the population into rapid growth. Those nutrients now include a source that scientists had not accounted for. The ammonium, produced by the nitrogen-fixing bacteria beneath the ice, dissolves into the water column over the winter and early spring. By the time the algae bloom season starts, a reservoir of ammonium has accumulated. When light returns and algae begin dividing, they have access to more nitrogen than the old models assumed was available. The result is a bloom that outruns predictions. Satellite ocean color sensors have tracked algae blooms across the Arctic for decades. These sensors detect the green pigment that algae produce during photosynthesis. The data show blooms appearing earlier in the season, extending later, and in some regions growing denser than models built from recycled nutrient assumptions would predict. Researchers have proposed several explanations for this pattern over the years: warmer water temperatures, more open water from ice retreat, changed circulation patterns bringing nutrients from deeper water to the surface. All of those factors are real and contribute to the shifting bloom patterns. The bacterial nitrogen contribution is now an additional explanation that fits the data and the timing. Ammonium accumulation under the ice through winter, followed by release into the water column as spring mixing begins, delivers a pulse of fertilizer to the algae exactly when they are starting to bloom. The effect is confirmed in regions where bacterial nitrogen fixation rates were highest. Sites near the ice edge where bacterial activity peaked also showed the strongest algae bloom signals in satellite imagery during the study period. The blooms matter beyond their size. A dense algae bloom absorbs carbon dioxide from the water around it as the algae perform photosynthesis. The more algae, the more carbon dioxide pulled from the water. As ocean water loses dissolved carbon dioxide, it draws more down from the atmosphere above to restore the balance. The ocean acts as a pump, pulling atmospheric carbon dioxide into the surface layer and then into the algae. This process is called the biological carbon pump, and it is one of the primary ways the ocean removes carbon dioxide from the atmosphere over timescales that matter for climate. A larger and more productive algae bloom means a stronger pump. The bacteria under the ice are feeding the pump without anyone having asked them to. Biology professor Lasse Riemann, who was involved in interpreting the Copenhagen team's findings, stated that increased algae production means the Arctic Ocean will absorb far more CO2 than current models predict. That statement carries weight. Current climate projections include ocean carbon absorption as a key variable. If the Arctic absorbs significantly more carbon dioxide than models assume, those projections shift. The direction of the shift is, on the surface, encouraging. More carbon dioxide absorbed means less warming. But the full picture is more complicated than that, and the mechanism that drives the carbon from the algae all the way down to the ocean floor is one of the stranger things biology does at scale. The ocean breathes. At the surface, carbon dioxide moves constantly between the air and the water. When the atmosphere has more carbon dioxide than the surface water, the gas dissolves down into the ocean. When the surface water has more, it releases carbon dioxide back into the air. The two stay in a rough balance, shifting with temperature, wind, and chemistry. Algae break that balance deliberately. During photosynthesis, algae pull dissolved carbon dioxide out of the water and lock it inside their cells as carbon-based organic compounds. These compounds build the algae's body, their cell walls, their internal structures, their stored energy. Every algae cell that forms represents carbon dioxide that has been removed from the water and converted into living matter. As algae remove carbon dioxide from the surface water, the water's carbon dioxide concentration drops. The atmosphere, still carrying its own carbon dioxide load, pushes more gas down to restore the balance. The ocean draws carbon dioxide out of the air to replace what the algae consumed. The algae act as a sponge. They soak carbon out of the water. The water then refills from the atmosphere. The net effect is a one-way movement of carbon dioxide from the air into the ocean, driven by biological consumption at the surface. In a small bloom, this effect is small. In a massive bloom covering thousands of square miles of Arctic Ocean, the absorption is significant enough to show up in atmospheric measurements. The connection to the bacteria runs directly through this mechanism. The bacteria produce ammonium. The ammonium fertilizes the algae. The fertilized algae bloom faster and larger. The larger bloom removes more carbon dioxide from the water. The water draws more carbon dioxide from the atmosphere. The net result is a larger Arctic carbon sink than models had calculated. Every part of that chain has now been confirmed in the basic biology. The nitrogen-fixing bacteria are real and active. The ammonium they produce is real and measurable. The relationship between ammonium availability and algae growth is well established. The relationship between algae photosynthesis and ocean carbon dioxide absorption is fundamental physics and chemistry. The scale of the effect is the part that remains a theory. Scientists cannot yet calculate the total annual carbon dioxide absorption boost the Arctic receives from this bacterial system. The calculation requires knowing the total bacterial nitrogen input across the full Arctic Ocean, a number that the 13 sampling sites have not yet resolved. What the biology confirms is the direction. The bacteria are making the Arctic a bigger carbon sink than it was understood to be. The ocean is absorbing more carbon dioxide than the models show. That shifts every downstream calculation that depends on ocean carbon absorption, including projections for how fast atmospheric CO2 concentrations will rise, and how quickly that rise will drive temperature increases. A single paper published in October 2025 changed the inputs to some of the most important equations in climate science. But the most dramatic part of this story is not what happens while the algae are alive. It is what happens when they die and where the carbon inside them eventually ends up. When an algae cell dies, it sinks. That is the core of one of the most important geological processes on Earth. And it is the final step in the chain that connects a bacterium producing ammonium under Arctic ice to the carbon dioxide concentration in the air you breathe. Here is the full path. A bacterium under the ice converts nitrogen gas into ammonium. The ammonium dissolves into the water column and rises toward the ice above. Algae absorb the ammonium and use it to accelerate their growth. The algae bloom. They pull carbon dioxide out of the ocean water, through photosynthesis and lock that carbon inside their cells. Then the algae die. Dead algae cells are slightly denser than seawater. They sink. As they sink, they carry the carbon stored inside them downward through the water column. Some of that carbon gets eaten by other organisms on the way down. Those organisms use the carbon for energy and release some of it back as carbon dioxide. But a significant fraction of the carbon inside dead algae cells makes it all the way to the ocean floor before being broken down completely. On the ocean floor, that carbon gets buried in the sediment. Sediment accumulates in thin layers over centuries and millennia. The buried carbon stays locked in those layers, removed from the ocean water, removed from the atmosphere, effectively stored. This process has been running continuously for hundreds of millions of years. It is the reason Earth's atmosphere has not accumulated runaway levels of carbon dioxide from volcanic eruptions and biological respiration. The ocean floor is a long-term carbon storage vault, and sinking algae cells are one of the main delivery mechanisms. The biological carbon pump is the scientific name for this system. Algae grow at the surface, absorb carbon, die and sink. The pump moves carbon from the atmosphere downward through the ocean and into the sediment below. The Arctic has always participated in this process, but the models built before the Copenhagen discovery assumed a much smaller algae base in the polar ocean than may actually exist, because they did not account for bacterial nitrogen fertilization. If the algae bloom in the Arctic is larger than models assumed, then the sinking carbon flux is also larger. More dead algae cells falling through the water column, more carbon reaching the sediment, more long-term storage. The bacteria are effectively running an unauthorized carbon removal operation at the top of the world. Unauthorized because no one planned it. No one engineered it. No government agency commissioned it. Millions of years of evolution produced a system that, as a byproduct of its own survival chemistry, removes carbon from the atmosphere and buries it in the ocean floor. This is an active process running right now, every day, under the ice. Biology professor Lasse Riemann described the bacterial system as something that will cause the Arctic Ocean to absorb far more CO2 than current models predict. That statement is a theory, carefully labelled as such by the researchers. The total magnitude of the effect remains unknown, but the mechanism is confirmed and the direction is established. The bacteria are making the Arctic a bigger carbon sink. The climate models do not know this yet. The gap between what the models show and what the bacteria are actually doing represents one of the most significant blind spots in current climate science. And that blind spot has been hiding in plain sight inside every climate projection produced for the last several decades. Climate models are built from data. Researchers collect temperature readings, gas concentrations, ocean chemistry measurements, biological activity rates, and hundreds of other variables. They feed these into computer simulations that project how the Earth's climate will change over years, decades, and centuries. These models are used to forecast sea level rise, temperature increases, and the pace of Arctic ice loss. Every major international climate report, including those used to guide government policy, energy investment, and infrastructure planning, draws from these models. The models have a problem now. A significant input was wrong. Every model that calculates how much carbon dioxide the Arctic Ocean absorbs was built using estimates of biological productivity in that ocean. Biological productivity in the Arctic Ocean depends on the availability of nitrogen, since nitrogen is the key limiting nutrient for algae growth. Nitrogen availability was estimated based on the assumption that nitrogen fixation in the Arctic Ocean was essentially zero. That assumption is wrong. Confirmed. The bacteria under the Arctic ice are adding nitrogen to the system. How much nitrogen, across the full Arctic Ocean, over a full year, remains an open calculation. Scientists do not yet have enough sampling points to extrapolate accurately from 13 sites to 5.5 million square miles of ocean. What they know is the direction, The nitrogen input is larger than zero. The biological productivity supported by that nitrogen is larger than models calculated. The carbon absorption driven by that productivity is larger than models predicted. When an input is wrong, every output downstream from it is also wrong. The models that show how much carbon dioxide the Arctic absorbs each year are off. The models that use that absorption number to calculate net atmospheric carbon dioxide are off. The projections that use those atmospheric numbers to estimate temperature rise are off. The question is how far off? At this stage, that answer is a theory. Researchers believe the effect is significant enough to matter to global projections, but the total scale has not been quantified. Quantifying it requires more expeditions, more sampling sites, more years of data. The research community is now mobilizing to fill that gap. There is also a secondary unknown. The models built without this bacterial nitrogen input generated predictions that policymakers used to set emissions targets, design climate adaptation plans, and negotiate international agreements. If those predictions underestimated the ocean's natural carbon absorption capacity, the gap has implications for how much human carbon emissions actually matter at the margins of those agreements. Climate scientists are careful to note that the bacterial carbon sink does not cancel out human emissions. The scale of human CO2 output, roughly 40 billion tons per year globally, is far larger than any reasonable estimate of what the Arctic bacteria might offset. But in a system where small shifts in the balance can cascade into large effects, a missing variable of this size cannot simply be incorporated into existing models by adjusting a single number. The models need to be rebuilt with new biology. That rebuilding process is underway, and while it proceeds, the bacteria continue their work. No meeting convened to authorize it. No scientist engineered it. They convert nitrogen in the dark, release ammonium, feed the algae, drive the carbon pump, and store carbon in the seafloor. The machinery that runs this has been operating for a very long time, and understanding how it actually works at the molecular level reveals something about these bacteria that is even more unsettling than their location. The water samples came up cold and dark inside sealed glass bottles. Inside those bottles, mixed with Arctic seawater, were billions of microscopic cells. Some were alive and active, some were dead, some were fragments of cells carried by currents from thousands of miles away. The challenge for the Copenhagen research team was figuring out which were which. Genetic sequencing answered that question definitively. The process begins with filtration. Researchers push the water sample through an extremely fine membrane that catches cells too small to see with the naked eye. The membrane traps the biological material while the water passes through. What remains on the filter is a concentrated layer of microscopic life from that specific depth and location. From that layer, scientists extract deoxyribonucleic acid, the full genetic blueprint of every organism present in the sample. Then they sequence it. Modern sequencing technology can read millions of genetic fragments simultaneously, identifying which genes are present and, critically, which ones are actively being expressed. A gene being present means an organism carries the instructions for a particular function. A gene being expressed means the organism is actively using those instructions right now, building the proteins that execute the function. The team searched specifically for NIF genes. These genes encode the proteins that make up nitrogenase, the enzyme complex that performs nitrogen fixation. Finding NIF genes in a sample only tells you those organisms passed through. Finding NIF genes actively expressed tells you the organisms are running a reaction at the moment of collection. Every site produced active NIF gene expression. Confirmed. This result eliminated the alternative explanation that worried the researchers most. Ocean currents carry biological material across enormous distances. A warm water cyanobacterium swept north by a current could end up in an Arctic sample carrying its NIF genes into an environment where it is completely inactive and dying. Its genetic material would still appear in a sequencing result, and could be misread as evidence of local nitrogen fixation. The gene expression data ruled this out. The cells in the Arctic samples were producing nitrogenase enzymes. Dead or dormant cells carried north by currents do not produce enzymes. The bacteria in the water below the Arctic ice were metabolically active, fixing nitrogen in real time at the moment the samples were collected. A second layer of genetic evidence reinforced this conclusion. The researchers mapped the full genomic profile of the dominant nitrogen-fixing organisms in their samples. The genome is the complete instruction manual of an organism, not just one set of genes. The Arctic bacteria showed a genomic profile that matched cold-adapted organisms, carrying specific gene variants that control membrane flexibility, cold-stable protein production, and energy management under low-nutrient dark conditions. These adaptations are not present in tropical nitrogen-fixing bacteria. They are features that develop over long evolutionary periods in cold, dark environments. An organism swept in from warmer water would carry a different genomic profile entirely. The Arctic bacteria looked like they belonged there, because they did. The genetic evidence confirmed what the chemical measurements showed. A resident community of nitrogen-fixing bacteria adapted over a long evolutionary history to survive and function in one of the harshest marine environments on Earth. And the specific molecular machinery that allows their enzymes to function at temperatures that freeze most biological chemistry turns out to operate through a mechanism that surprised the researchers examining it. Enzymes are proteins. Proteins are chains of amino acids folded into specific shapes. The shape of a protein determines its function. When a protein gets too cold, the chain stops moving freely. The molecule becomes rigid. The active site, the specific region of the protein that performs its chemical work, locks up and stops functioning. This is why most biological chemistry slows dramatically in cold temperatures and halts entirely near or below freezing. Nitrogenase, the enzyme that fixes nitrogen, is large and complex. It has two main protein components that must work together in sequence. One component captures nitrogen gas and holds it in place. The other delivers electrons to break the nitrogen gas apart and attach hydrogen atoms, creating ammonium. The two components must repeatedly connect and separate in a precise choreography to complete the reaction. At tropical ocean temperatures, around 77 degrees Fahrenheit, this choreography runs smoothly. The proteins are flexible enough to connect, react, and separate thousands of times per second. At 28 degrees Fahrenheit, the standard version of nitrogenase found in tropical bacteria would seize. The proteins would lose the flexibility needed for their components to connect properly. The reaction would stop. The Arctic bacteria run a modified version. Their nitrogenase proteins carry specific amino acid substitutions at key positions throughout the molecule. These substitutions do not change the basic function of the enzyme. The active site still captures nitrogen, the electron delivery component still fires, but the substitutions change the physical properties of the protein chain in ways that preserve flexibility at sub-zero temperatures. The most important change involves the hydrophobic core of the protein. In most proteins, a cluster of water-repelling amino acids folds into the interior of the molecule and holds its three-dimensional shape stable. In cold environments, that core can become too rigid, locking the protein into a shape that cannot flex through its functional range of motion. The Arctic bacteria use amino acids with smaller, less rigid side chains in some of these core positions. The result is a protein interior that remains somewhat flexible even as temperatures drop toward freezing. The enzyme can still perform its connection and separation choreography, just at a slower rate than it would in warm water. This is a confirmed feature of cold-adapted enzymes found across multiple polar organisms. Marine bacteria in Antarctic waters show similar modifications. Certain Arctic fish produce antifreeze proteins using related structural principles. The Arctic nitrogen-fixing bacteria apply the same cold adaptation logic to one of the most energetically demanding enzyme systems in biology. The trade-off is speed. Cold-adapted enzymes run slower than their warm water counterparts at any given temperature. The Arctic bacteria fix nitrogen at a lower rate per cell than tropical cyanobacteria do. They compensate by running continuously, maintaining bacterial communities dense enough that their collective output produces measurable nitrogen fixation across the water column. Quantity of time replaces intensity of reaction. A slower engine running without pause produces real output. The metabolic pathway supporting all of this, the system that extracts energy from dissolved organic matter to power the nitrogenase reaction, carries its own cold adaptations. The membrane through which the bacteria absorb organic material stays fluid at temperatures that would solidify a typical bacterial membrane. The whole organism is engineered, by evolution, for exactly this environment. Understanding how this engineering developed requires looking back at a gap in the scientific record that nobody thought to investigate until very recently. The first signal appeared in 2017, buried inside a dataset that nobody initially flagged as remarkable. Marine chemists studying nutrient cycles in polar waters noticed that nitrogen levels in certain Arctic Ocean regions did not match what the standard recycling model predicted. The water carried slightly more bioavailable nitrogen than it should have if the only source was decomposed organic matter from above. The numbers were small. The discrepancy was easy to dismiss as measurement error or local variation caused by currents mixing in water from elsewhere. Nobody published a paper specifically about the nitrogen anomaly. The data sat in the background, noted and moved past. In 2019, a different research group working on cold ocean biology identified symbiotic cyanobacteria operating in cold surface waters near polar regions. These were sunlight-dependent nitrogen fixers, meaning they still fit within the existing model framework. They used photosynthesis. They lived in the surface layer where some light reached them. The discovery was interesting but incremental. It extended the known range of photosynthetic nitrogen fixation slightly poleward without overturning anything fundamental. The community filed it and moved on. The problem was instrumentation. Detecting low-level nitrogen fixation in cold, nutrient-sparse water requires sensitive equipment. The older tracer methods used on most polar expeditions before 2020 had detection limits that sat above the actual fixation rates occurring in the Arctic. The measurements came back near zero because the tools could not see below their own threshold. The Arctic appeared to produce almost no nitrogen fixation, so researchers designed their expeditions and their models around that appearance. The absence of a signal was treated as confirmed zero rather than as a potential measurement gap. The acetylene reduction and isotope tracing methods used by the Copenhagen team in 2023 had finer resolution. They could detect fixation rates at the low concentrations actually present in the Arctic water column. The difference in sensitivity between the older and newer methods is roughly comparable to the difference between a thermometer that reads to the nearest tenth of a degree. Both give you a temperature. Only one gives you the detail you need. The 2017 anomaly might have been resolved years earlier with better tools. The research community did not lack curiosity. It lacked the right equipment in the right place at the right time, combined with funding for an expedition specifically designed to answer the nitrogen question. Funding for polar research has historically trailed behind funding for tropical ocean work. The logistical cost of Arctic expeditions, requiring icebreaker ships and extended travel to remote regions, makes every data point more expensive. Researchers seeking grants often face pressure to choose questions that seem more accessible. The nitrogen anomaly in 2017 was a signal. Science's infrastructure at the time was not optimized to chase it. By 2023, the equipment had improved. A team had secured access to 2 major icebreakers, and the question had aged into urgency. Climate change was accelerating changes in the Arctic faster than any previous model had predicted. Researchers needed a better picture of the polar ocean's biological baseline before it changed further. The IB Odin and the RV Polarstern sailed into the Arctic with that urgency behind them. What happened when the first sample results came in was a moment that researchers on board will likely describe for the rest of their careers. The expedition launched in the summer of 2023. The IB Odin departed first, navigating north through the Norwegian Sea and into the Barents Sea before pushing into the pack ice of the central Arctic Ocean. The RV Polarstern followed a parallel route into the Eurasian Arctic Basin. Together, the ships covered a sampling grid designed to capture data across the widest possible range of ice conditions and geographic positions. The first sampling sites were near the Svalbard archipelago, in the waters north of that Norwegian island group, where the ice edge sits during summer. The researchers lowered their collection equipment and brought up their first water samples. The samples went through the laboratory processing protocol: filtration, genetic extraction, chemical tracer incubation. The teams worked through the night on their first samples, running the acetylene reduction assays and preparing the genetic material for sequencing. The results from Site 1 showed active nitrogen fixation, measurable, above the detection threshold. Real. The team ran the controls again. Controls are samples processed identically to the real samples, but using water treated to kill all biological activity. If the nitrogen fixation signal appeared in the controls, it meant contamination or instrument error. The controls showed nothing. The signal was biological, and it was coming from the living cells in the Arctic water. Lisa von Friesen, the lead researcher on the study, later described the moment as one where the team realized that everything they thought they knew about this ocean had to be reconsidered. The first positive result did not prove a systemic phenomenon, but it demanded they keep looking. They kept looking across 600 more miles of ocean. Every subsequent site produced the same result. Some sites showed lower rates. Sites near the ice edge showed higher rates. The geographic spread ruled out local anomaly. By the time the ships reached their final sampling locations northeast of Greenland, the team had a dataset covering 13 sites, each one confirming the pattern. The scientific paper that came out of this expedition, published in Communications, Earth and Environment on October 19, 2025, represented 2 years of analysis between the expedition and publication. The team spent those 2 years verifying the results, checking for alternative explanations, comparing their Arctic genetic sequences against global databases of nitrogen-fixing organisms, and calculating the fixation rates from their tracer data. Every check supported the original finding: active nitrogen fixation by non-cyanobacterial bacteria in waters previously assumed to be biologically inactive for this process. The discovery held across methods, across sites, and across the 2 years of post-expedition analysis. The paper landed in the scientific community like a signal that could not be ignored. News coverage spread within a day of publication. Researchers in marine biology, climate science, and polar ecology began reading the methods section to understand exactly how the Copenhagen team had detected what others had missed. The answer, as researchers across the field quickly recognized, came down to tools, targeting, and the willingness to assume that an unanswered question was genuinely unanswered rather than already settled. The personal stakes for the scientific community were significant. Careers and models had been built on the assumption that the Arctic was nitrogen neutral. That assumption was now confirmed wrong. But the most unsettling questions raised by the discovery were not about what had already been found. They were about what had not been looked for yet. And one particular gap in the data pointed toward a possibility that nobody had seriously proposed before. Lisa von Friesen is a marine microbiologist at the University of Copenhagen. She designed the sampling protocol for the 2000-2003 expedition. She led the analysis of the genetic data. She authored the paper that announced the discovery to the world in October 2025. When the first confirmed results came back from the Arctic samples, von Friesen's response was not immediate celebration. It was deep discomfort with what the numbers implied about the existing scientific framework. She stated publicly that science had been completely wrong about the boundaries of where these organisms could operate. That is a significant thing for a researcher to say. Scientific papers are carefully worded. Public statements from scientists about their field being completely wrong are rare. They carry weight precisely because scientists are trained to avoid overstatement. The discomfort she described came from a specific place. The assumptions that placed the limit of nitrogen fixation at warmer, sunlit waters were not casual guesses. They were built on decades of research, tested across multiple ocean environments, supported by the known biochemistry of nitrogenase enzymes. The textbook position had evidence behind it. Serious, accumulated, peer-reviewed evidence. Finding that the Arctic bacteria had evolved around those constraints did not mean the previous research was careless. It meant that the previous research had sampled the organisms that fit the existing model and missed the ones that did not. A model built only from what you can see is only as complete as your field of vision. Von Friesen's team had used better tools, sailed to a harder-to-reach location, and asked a question that the field had mostly treated as already answered. The result exposed a gap that had been invisible precisely because the methods used to look for it were not sensitive enough to find it. Her public statement about science being wrong was an acknowledgement of how gaps form in scientific knowledge. A community builds models from available data. The models become the default assumption. The default assumption shapes what questions seem worth asking. Questions that fall outside the assumption stop getting asked, even when the evidence for them begins to accumulate. The 2017 nitrogen anomaly was an early hint that the assumption might be wrong. Von Friesen and her team built an expedition specifically to test that hint with better tools. Biology professor Lasse Riemann, who contributed to the study's interpretation, framed the implications differently. He focused on the forward-facing consequences rather than the retrospective gap. His emphasis was on what the Arctic Ocean would now do differently in climate models, specifically its carbon absorption capacity once the bacterial nitrogen contribution was properly included. Both perspectives are essential. Von Friesen's discomfort acknowledges the cost of the blind spot in past science. Riemann's forward framing describes the urgency of correcting for it. What their combined statements describe is a scientific community at an inflection point. The discovery is made, the confirmation is solid, the next phase is quantification. How much nitrogen? Across how much area? In what seasonal pattern? Over what time period? Those questions drive the next generation of expeditions. And while scientists prepare to answer them, the mystery at the center of the whole system remains untouched. Nobody knows the total annual output of these bacteria for the entire Arctic Ocean. That number is one of the most consequential unknowns in current climate science, and the scientists searching for it face a problem they did not anticipate. 13 sites. 600 miles. The sampling is the best available evidence. It is nowhere near enough to calculate the total. The Arctic Ocean covers roughly 5.5 million square miles. The 13 sampling sites from the 2023 expedition represent a fraction of that area so small it is difficult to express meaningfully. Imagine trying to map the soil chemistry of the entire continental United States using samples from 13 locations spread across a 600-mile stretch from New York to Pittsburgh. You would have real data. You would not have a complete picture. The nitrogen fixation rates measured at those 13 sites ranged from 0.4 of a nanomole per liter per day in the central pack ice to 5 nanomoles per liter per day near the ice edge. That is a tenfold difference across sites, driven by ice conditions, algae density, and organic material availability. To extrapolate a total, researchers need to know what the rates look like across all the conditions present in the Arctic Ocean, not just the ones sampled on one expedition. That requires more expeditions, more sites, more years of data. The calculation is also time-dependent. Bacterial nitrogen fixation in the Arctic follows the seasonal cycle of algae. It accelerates during spring and summer when algae are most active and organic material is most abundant. It slows through the polar winter. The 2023 expedition captured a summer snapshot. Winter rates are unmeasured. Measuring winter rates in the Central Arctic Ocean requires working through polar night in total darkness, through ice that reaches its maximum thickness. That is a logistically extreme proposition. The icebreakers that supported the initial expedition are among the few vessels capable of operating in those conditions year-round. Access to those ships is limited and expensive. There is also a depth problem. The sampling on the expedition focused on the upper water column, the region directly beneath the ice, and extending down through the zone of known bacterial activity. The deep Arctic Ocean below 1,000 feet was not sampled for nitrogen fixation. Whether similar bacteria operate in the deep water column remains an open question. Scientists are working on modeling approaches to estimate the total annual nitrogen input from available data. These models use the measured rates as anchor points and apply ocean circulation data, ice condition maps, and algae productivity estimates to interpolate values across unsampled areas. The results so far suggest the total is substantial, large enough to matter to climate projections, but the range of uncertainty in those estimates is wide. Wide uncertainty means the models cannot be trusted to guide precise policy decisions. The difference between the low end and high end of current estimates for total Arctic nitrogen fixation is large enough to significantly change projections for Arctic carbon absorption capacity. Closing that gap requires a coordinated multi-year sampling campaign covering all seasons, all depth zones, and a geographic spread far wider than the initial expedition covered. The scientific community recognizes the urgency. The funding and logistical infrastructure needed to support such a campaign are still being assembled. Every year that passes without that data is another year that climate models operate with a missing variable. The models drive decisions. The decisions have consequences. A gap in the data is not just an academic problem. And while scientists scramble to measure the total nitrogen input, another question has emerged that is even harder to answer. One that requires looking not into the water below the ice, but into the ice itself. The water beneath the ice is cold and dark. The bacteria discovered there survive conditions that defeated scientific expectation. The ice itself is colder, darker, and more extreme. Arctic sea ice is not a solid, uniform block of frozen water. At the microscopic level, it is threaded with tiny channels called brine channels. As seawater freezes, salt gets pushed out of the forming ice crystals. The brine inside them can be many times saltier than regular seawater. The temperatures inside the channels can reach far below the freezing point of fresh water, sustained in liquid form only by the extreme salt concentration. Brine channels are measured in hundredths of an inch across. They run through the ice in networks, connecting to each other and sometimes to the water below. In warmer parts of the ice, near the underside, the channels are wider and more connected. Deeper into the ice, closer to the frozen surface above, they narrow and isolate. Microbial communities have been found living inside brine channels in sea ice before. These are typically algae and bacteria adapted to the intense salt concentration and cold. They survive on whatever organic material washes into the channels and on whatever light filters through the ice above them. The question the Copenhagen discovery raises is whether nitrogen-fixing bacteria specifically exist inside the brine channel system. If the non-cyanobacterial diazotrophs can function in the water below the ice at sub-zero temperatures, could a version of the same or similar organisms operate inside the ice matrix itself? The answer is currently unknown. Nobody has looked. The sampling methodology used on the expedition targeted water below the ice, not ice cores. Drilling ice cores for microbial analysis requires different equipment and different processing protocols. The genetic sequencing and nitrogen fixation assays used on water samples require adaptation to work on ice-extracted material, which has different physical and chemical properties. If bacteria are fixing nitrogen inside the brine channel network, The implications branch in two directions. First, it would expand the total nitrogen input estimate. Upward, brine channel communities and sea ice cover an enormous surface area across the Arctic. Even low fixation rates across that area would add meaningfully to the total. Second, it would push the boundaries of where life can survive even further than the water column bacteria already have. Life operating inside the matrix of frozen ocean, surrounded by ice, sustained only by brine chemistry and whatever organic material the channels contain, would represent an adaptation extreme even by the standards of the bacteria already discovered. This remains a theory carefully framed as such by the researchers. The existing data provide no direct evidence for or against nitrogen fixation inside the ice. It is a logical next question, not a confirmed observation. Testing it requires targeted ice core sampling on future expeditions, with analysis protocols specifically designed to detect low-level nitrogen fixation in brine-extracted samples. That work has not yet begun. The possibility hangs open. And whether bacteria live inside the ice or only in the water below it, what they are doing to the ecosystem above them is now rippling through food chains in ways that marine biologists are only beginning to track. The Arctic food web runs in layers. Bacteria and algae form the base. Zooplankton eat the algae. Small fish eat the zooplankton. Larger fish, seabirds, seals, and whales eat the small fish. The whole pyramid rests on the productivity of that lowest layer, the microscopic biology that converts sunlight and chemistry into living matter. When the base expands, every layer above it has the potential to grow. The bacterial nitrogen input confirms that the base of the Arctic food web has been receiving a fertilizer supply that models did not account for. Ammonium from nitrogen-fixing bacteria feeds algae. More ammonium means more algae. More algae means more food available for the zooplankton that depend on them. Zooplankton are the link between the microscopic and the visible. Species like copepods, tiny crustaceans smaller than a grain of rice, exist in enormous numbers in Arctic waters. They graze on algae continuously, converting the energy stored in algae cells into animal tissue. That tissue feeds the fish above them. Arctic cod is one of the most important fish in the polar food web. They are a primary prey species for ringed seals, beluga whales, and numerous seabirds, including thick-billed murres. Arctic cod feed heavily on copepods. Their population size tracks closely with zooplankton availability. More zooplankton means more cod. More cod means more of everything that depends on them. If bacterial nitrogen fixation is increasing the Arctic algae base, the effect should propagate upward through copepods and into the populations of marine mammals and seabirds that define the Arctic ecosystem. This is a theory. The data confirming bacterial nitrogen fixation are solid. The cascade through the food web is a logical projection based on well-established ecological relationships, but it has not been directly measured as a consequence of the bacterial discovery. Scientists flag this clearly. The complication is that climate change is simultaneously reshaping the Arctic food web through other mechanisms. Warming waters are expanding the range of subarctic fish species northward. Pacific cod and capelin are moving into territory previously occupied only by Arctic-adapted species. These newcomers compete with Arctic cod for zooplankton and prey on the same fish. A larger algae base, boosted by bacterial nitrogen, could support both the expanding population of resident Arctic species and the incoming subarctic competitors, or the newcomers could outcompete the residents even in a more productive system. The interactions are complex. The expanded food base also has implications for human communities that depend on Arctic marine life. Indigenous communities across northern Alaska, Canada, and Greenland have sustained themselves on Arctic marine mammals and fish for thousands of years. Changes in the distribution and abundance of those species affect food security, cultural practice, and community health. Commercial fishing interests are also tracking the northward shift of fish populations. As more productive waters open up in the Arctic, fishing pressure from international fleets increases. The bacterial discovery adds a layer of biological complexity to an already politically charged set of questions about who has access to Arctic marine resources and how those resources should be managed. The microbes at the bottom of the food chain are connected through a chain of organisms and consequences to geopolitical negotiations at the top of the world. And the connection between those microbes and the world outside the Arctic runs through a pathway that reaches far beyond fish populations. Your city's temperature is connected to a bacterium you will never see, living in a place you will likely never visit. The connection is indirect, but real, and it runs through carbon dioxide. The atmosphere holds carbon dioxide like a blanket holds heat. More carbon dioxide in the atmosphere means more heat retained near the surface. Global average temperatures rise. Ice melts faster, weather patterns shift, heat waves intensify, coastal flooding increases. The specifics vary by region, but every part of the planet is tied to the global atmospheric carbon dioxide concentration. The Arctic Ocean is one of the places that helps regulate that concentration. Cold water absorbs carbon dioxide more readily than warm water does. The Arctic Ocean has historically been a significant carbon sink, pulling carbon dioxide from the atmosphere and storing it in the water column and eventually in seafloor sediment through the biological carbon pump. Climate models assign the Arctic Ocean a specific carbon absorption value. That value feeds into calculations of how fast atmospheric DOC rises. Those calculations inform temperature projections. Temperature projections drive policy decisions about emissions targets, infrastructure investment, and adaptation spending. The bacterial nitrogen input changes the Arctic's carbon absorption value upward. By feeding the algae bloom that drives the biological carbon pump, the bacteria cause the Arctic to absorb more carbon dioxide than the models calculated. The atmospheric concentration of carbon dioxide rises slightly more slowly than models project because the Arctic is pulling more of it into the ocean than expected. The effect is real. Its magnitude is still being calculated. Scientists have confirmed the mechanism and established the direction but have not yet quantified the total annual difference in carbon absorption. Even a modest increase in the Arctic's carbon absorption capacity matters at the margins of the climate system. The difference between 1.5 degrees Celsius of global warming and 2 degrees Celsius of warming sounds small. It corresponds to hundreds of millions of people exposed to severe heat stress, entire island nations losing habitability, and coral reef systems moving from damaged to functionally extinct. The margins matter. A carbon sink the size of the Arctic performing above its modeled capacity could translate into years of additional time before critical temperature thresholds are crossed. That is a theory based on the confirmed biology and awaiting quantification. But the direction of the effect is established. The bacteria are providing a service to the climate system entirely by accident. They fix nitrogen because it allows them to survive. They have done this for millions of years with no relationship to human civilization and no awareness of the atmospheric chemistry they influence. The consequence of their survival chemistry, across billions of individuals operating continuously under the ice, is a measurable shift in one of the most important physical parameters of the planet. Your city's summer temperatures, your region's rainfall patterns, the severity of the storm season you experienced this year, are all downstream of a chain that includes these bacteria in its calculations, whether the models know it yet or not. Understanding what humanity owes to these invisible organisms begins with understanding why scientists spent so long dismissing the environment where they live as biologically empty. The cost of that assumption runs deeper than a few incorrect numbers in a climate model. The term biological desert does not mean nothing lives there. It means the productivity is so low, the biological activity so sparse, that the environment contributes almost nothing to regional nutrient cycles. A biological desert is a placeholder, a zone where models assign near zero biological input and move on. The Arctic Ocean earned that label gradually, through a combination of difficult access, limited tools, and a scientific culture that rewarded expeditions to more accessible and apparently more active environments. The label stuck because nothing appeared to contradict it. The tools available for most of the 20th century were not sensitive enough to detect what was actually happening in the cold, dark water. The cost of the label accumulates over decades. Every climate model built since the 1980s included an ocean carbon absorption calculation. The calculation depended on estimates of biological productivity in different ocean regions. The Arctic's biological productivity was set low, reflecting the desert assumption. Models ran forward from that baseline, projecting future atmospheric carbon dioxide concentrations and temperature trajectories. International climate negotiations used those projections. The Paris Agreement targets of limiting warming to 1.5 and 2 degrees Celsius were set using models that did not include Arctic bacterial nitrogen fixation. Emissions reduction pathways, the plans that countries use to reduce greenhouse gas output fast enough to hit those targets, were designed around the modeled behavior of the climate system, including the modeled behavior of the Arctic Ocean. If the Arctic Ocean absorbs significantly more carbon dioxide than the models assumed, the remaining carbon budget, the total amount of carbon dioxide humanity can still emit while staying within the temperature targets, changes. The direction of that change is an increase in the remaining budget. The system is absorbing more than projected. This sounds like good news. The problem is that relying on an unquantified sink to extend a carbon budget is not sound policy. The total bacterial nitrogen input and its effect on Arctic carbon absorption remain unquantified. Building emissions plans around an uncertain natural process that could change as the ice continues to retreat introduces new risks. The bacteria's productivity depends on ice. They need the algae that grow on and near the ice to provide their fuel. As the Arctic warms and ice coverage shrinks further, the relationship between ice extent and bacterial productivity may change in ways that are difficult to predict. More melt edge in the short term increases bacterial habitat. Complete ice loss in the long term removes the algae communities the bacteria depend on. An ocean that no longer has summer ice is a fundamentally different biological environment from one that retains some ice coverage. The nitrogen-fixing bacteria adapted to the current Arctic, with ice present. How they behave in an ice-free Arctic summer is unknown. The biological desert assumption cost climate science decades of incomplete modeling. The cost going forward is less clear because the variable that was missing from the model is itself changing along with the climate. The scientists trying to correct for the missing variable are chasing a moving target. And the search for similar hidden biological systems in other extreme environments on Earth has just become significantly more urgent. The Arctic discovery changed one specific assumption, that nitrogen fixation requires warmth and light. That assumption did not only apply to the Arctic, it shaped how scientists thought about dozens of other cold and dark environments across the planet. Those environments are now being reconsidered. The deep ocean is the largest cold and dark environment on Earth. Below 3,000 feet, sunlight never reaches. Temperatures hover near freezing year-round. The pressure is crushing. Organisms living there have evolved in complete isolation from the surface world, running entirely on organic material that sinks from above or on chemical energy from hydrothermal vents. Deep ocean environments contain enormous volumes of dissolved nitrogen gas. If non-cyanobacterial diazotrophs can operate under Arctic ice in the dark at sub-zero temperatures, the question becomes whether related organisms exist in the deep ocean, fixing nitrogen in the cold and pressure of the abyssal zone. Early research had assumed the deep ocean contributed very little to global nitrogen fixation, for the same reasons the Arctic was dismissed. Too cold, too dark, too energy poor to sustain the expensive nitrogen-fixing reaction. The sensitivity limits of older tools may have produced the same measurement illusion in the deep ocean that they produced in the Arctic. Testing this requires deep ocean sampling with the same high-sensitivity methods the Copenhagen team used in the Arctic. Some preliminary work has already been done in deep Atlantic and Pacific waters, and hints of nitrogen fixation have appeared at depths previously considered too extreme. Those results are preliminary and not yet confirmed to the same standard as the Arctic findings. Antarctic waters present a similar question. The Southern Ocean surrounding Antarctica is cold, productive in certain seasons, and covers an enormous area. If nitrogen-fixing bacteria operate under Arctic ice, the waters surrounding Antarctic ice shelves and sea ice are logical candidates for the same investigation. Antarctic marine research has intensified in recent years, but targeted nitrogen fixation sampling with modern tools has not yet been as systematic as the Copenhagen expedition. High-altitude lakes frozen for most of the year represent another environment where the assumption of biological inactivity in cold, dark conditions has historically guided research. Some of these lakes contain liquid water year-round under their ice covers, in conditions that share characteristics with the Arctic under-ice zone. The broader principle is now in play. Cold, dark, seemingly hostile environments that science labeled low productivity or biologically inactive based on older tools and framework assumptions should be resampled with modern methods. The Arctic discovery suggests that the number of environments where hidden biology operates at scales that matter to global nutrient cycles may be larger than the existing scientific map shows. Each re-examination requires significant resources. Expeditions to deep ocean sites, Antarctic waters, and remote high-altitude lakes are expensive and logistically complex. The scientific community must prioritize which environments offer the most information per dollar of research investment. The Arctic discovery has shifted the calculation. The potential payoff of finding another hidden nitrogen cycle in a major ocean region is now clearly large enough to justify significant investment. The most dramatic version of that search, however, does not take place on Earth at all. Europa is Jupiter's fourth largest moon. It is slightly smaller than Earth's moon and covered entirely by a shell of ice. Under that ice shell, liquid water exists. Confirmed by magnetic field measurements and surface geology observed by spacecraft, the ocean beneath Europa's ice is estimated to be between 40 and 100 miles deep, containing more liquid water than all of Earth's oceans combined. The ice shell above it varies in thickness from a few miles to possibly tens of miles. No sunlight penetrates to the ocean below. The environment is dark, cold, and isolated. For decades, the search for life on Europa focused on the question of energy. Without sunlight, how would life survive? The main candidate was chemical energy from hydrothermal vents on the ocean floor, where Europa's internal heat, generated by gravitational tides from Jupiter, might drive the same kind of chemistry found in Earth's deep sea vents. Life near those vents would not need sunlight at all. The Arctic bacterial discovery adds a second framework. The non-cyanobacterial diazotrophs under Arctic ice do not use sunlight. They use dissolved organic carbon from dead algae. On Europa, there are no algae to produce dissolved organic carbon in the ocean, but the ice shell itself may contain organic compounds delivered by meteorite impacts over billions of years. Tidal flexing of the ice could drive mixing that delivers those compounds downward into the ocean. If the energy source available in Europa's ocean includes dissolved organic compounds, even at low concentrations, the Arctic discovery shows that bacteria can run the most expensive biological reactions, including nitrogen fixation, on exactly that kind of fuel. The conditions that scientists previously thought made Arctic water uninhabitable for nitrogen fixation, cold, dark, dissolved organic matter dependent, are precisely the conditions the Arctic bacteria thrive in. Europa's ocean shares at least some of those characteristics. This does not mean life exists on Europa. The leap from these bacteria survive in cold, dark water to therefore life exists in another planet's moon is enormous, and scientists are careful to avoid making it directly. But the discovery shifts the probability assessment. It removes one more constraint on where life can operate. It expands the chemical and physical parameter space within which biology is confirmed to function. The same logic applies to other icy moons. Enceladus, a moon of Saturn, vents water vapor and organic compounds from cracks in its ice shell, confirming a liquid water ocean below. Titan, another Saturn moon, has lakes of liquid methane on its surface and a complex organic chemistry in its atmosphere. Ganymede and Callisto, both moons of Jupiter, likely carry subsurface oceans as well. Every icy world with liquid water beneath a frozen shell is now a slightly more plausible candidate for biology, in light of what the Copenhagen team found beneath the Arctic ice. The bacteria under Earth's ice expanded the definition of habitable. The universe contains a very large number of cold, dark, icy environments with liquid water below. The search for life in those environments is now operating with a wider set of biological precedents. And back on Earth, a question about the long-term survival of the Arctic bacteria themselves raises an issue that nobody had considered when the discovery was first announced. The bacteria need the ice. They do not live in the ice directly, they live in the water below it. But they depend on the ice for everything that sustains them. The algae that grow on the underside of the ice and in the meltwater near the ice edge produce the dissolved organic carbon that the bacteria consume. Remove the ice, remove the algae that depend on it, and you remove the fuel supply for the bacteria. An ice-free Arctic summer would be a fundamentally different environment. Without ice cover, sunlight would reach the surface of the ocean directly. Algae would still grow, but in open water conditions rather than under ice conditions. Open water algae communities are different from ice algae communities. They are dominated by different species, grow in different patterns, and produce different kinds and amounts of organic material. Whether the non-cyanobacterial diazotrophs can adapt to consuming the organic material produced by open water algae communities is unknown. The bacteria are adapted to the specific chemistry of under-ice organic matter, the particular sugars, proteins, and dissolved compounds produced by ice algae under low-light, cold conditions. Open water algae produce different compounds in different proportions. This is not a confirmed barrier. It is a biological uncertainty. Bacteria adapt. Natural selection operates on bacterial populations across enormous numbers of individuals and relatively short generational timescales. But the speed of Arctic ice loss driven by current climate change may outpace the ability of the bacteria to adapt their metabolic preferences. If the bacteria cannot adapt quickly enough, and if ice-free Arctic summers arrive before they adjust, their populations could decline sharply. A declining bacterial population means declining nitrogen input. Declining nitrogen input means smaller algae blooms. Smaller algae blooms mean less carbon dioxide absorbed by the Arctic Ocean. The carbon sink shrinks. The same warming that initially expanded the ice edge and boosted bacterial activity could, if it continues far enough, destroy the habitat the bacteria depend on and reverse the enhancement. This creates a potential tipping point in the Arctic carbon system. Moderate ice loss increases bacterial activity and carbon absorption. Severe ice loss removes the biological infrastructure that supports that activity and reduces carbon absorption below current modeled levels. The curve of Arctic carbon absorption, plotted against temperature rise, may rise before it falls. Scientists flag this as a theory requiring further research. The data confirming bacterial nitrogen fixation say nothing directly about what happens to those bacteria in an ice-free summer scenario. That experiment has not occurred yet in the observable record. The Arctic is projected to experience its first ice-free September before 2050, based on current climate trajectories. That projection comes from models that did not include bacterial nitrogen fixation. Updated models that incorporate the bacteria may revise both the carbon absorption trajectory and the consequences of ice loss for biological productivity. The timeline for that revision is urgent. The window between now and a potential first ice-free Arctic summer is measured in decades, and the decisions made about carbon emissions in that window will shape which scenario unfolds. The bacteria evolved for a specific Arctic. That Arctic is changing. How far it changes depends on choices made far from the frozen ocean where the bacteria do their invisible work. Evolution does not keep records. The genetic code inside a living organism is the accumulated result of billions of years of mutation and selection, but it does not come labeled with dates. Researchers can compare genomes across species and estimate when lineages diverged based on the rate at which mutations accumulate over time. But those estimates carry wide uncertainty ranges, especially for microbial evolution, where horizontal gene transfer, the swapping of genes between unrelated organisms, complicates the family tree. For the Arctic nitrogen-fixing bacteria, the evolutionary history is genuinely unclear. The core nitrogen-fixing genes, the NIF gene cluster encoding nitrogenase, are ancient. They predate the rise of atmospheric oxygen on Earth, which places their origin somewhere beyond 2.5 billion years ago. The basic machinery of nitrogen fixation is one of the oldest biological functions known to science. Organisms have been fixing nitrogen in various forms since the early ocean. The cold and dark adaptations visible in the Arctic bacteria are a different question. Those are later additions to an ancient framework. At some point in the evolutionary history of these microbes, ancestors living in warmer conditions moved into colder environments, or cold environments expanded around populations that were already there, and the survivors carried genetic variants that allowed them to function at lower temperatures. The modifications to nitrogenase proteins that allow cold temperature function represent significant evolutionary work. Changing the amino acid composition of a critical enzyme without destroying its function requires many sequential mutations, each one either neutral or slightly beneficial, selected across thousands of generations. How long this took is unknown. Whether it happened once in a single ancestral lineage and then spread to related organisms through horizontal gene transfer is unknown. Whether the cold adaptation evolved independently in multiple separate bacterial lineages, which would be a striking example of convergent evolution, is also unknown. The genetic divergence between the Arctic bacteria and their tropical relatives suggests substantial evolutionary distance, substantial enough to imply either a very long time in cold conditions or very strong selective pressure that accelerated the pace of change, or both. Scientists have proposed, but not confirmed, that the ancestors of these bacteria may have colonized cold environments during earlier periods of Earth history when the planet experienced more extensive ice coverage. Periods called snowball Earth events, roughly 700 million years ago, froze much of the planet's ocean surface. Organisms that survived those events in pockets of liquid water would have faced intense selective pressure for exactly the cold adaptations the Arctic bacteria now carry. This is a theory. Testing it would require ancient DNA analysis from sediment cores, covering periods long before the current Arctic ecosystem took shape, and would involve analytical challenges that push the limits of current paleogenomics. The missing link in the evolutionary story is not just an academic curiosity. Understanding how these bacteria evolved to their current capabilities tells researchers something about how quickly biological adaptation can respond to environmental change. That question is directly relevant to whether the bacteria can adapt fast enough to keep up with the rate of change the Arctic is currently experiencing. The answer may determine whether the biological engine running under the ice survives the transformation of the world above it. And one final piece of the story connects everything that has happened since the discovery to a challenge that will shape Arctic science for the next generation. One paper, 72 pages in the published version of the journal, 4 weeks from submission to peer review acceptance, 2 years from expedition to publication. The paper appeared in Communications Earth and Environment on October 19, 2025. Within 24 hours, News coverage had reached every major science publication on the planet. Within a week, climate modeling groups in the United States, Europe, and Asia had begun internal discussions about how to incorporate the findings. Rewriting a climate model is not a simple task. Global climate models are among the most complex pieces of software humans have ever built. They simulate the physics of the atmosphere, the chemistry of the ocean, the movement of ice sheets, the exchange of gases at every surface, and the biological activity of every major ecosystem on Earth. These models run on supercomputers for days or weeks to produce a single projection. Thousands of researchers have contributed code, validation data, and calibration work over decades. Inserting a new variable requires more than adding a number. The bacterial nitrogen input to the Arctic Ocean affects biological productivity, which affects algae growth, which affects carbon dioxide absorption, which affects atmospheric carbon dioxide concentrations, which affects temperature and precipitation patterns globally. Every downstream effect of changing the Arctic biological productivity estimate ripples through interlinked components of the model. The first challenge is parameterizing the new variable. Climate modelers need a number, specifically a range of numbers representing the seasonal and geographic variability of bacterial nitrogen fixation across the Arctic Ocean. The 13 sampling sites from the 2023 expedition give anchor points, but cover far less than 1% of the ocean area that needs to be characterized. Modelers are currently working from extrapolations based on ice condition maps, ocean color satellite data, and the relationships observed in the expedition data between algae density and bacterial activity. These extrapolations carry wide uncertainty ranges. Wide uncertainty in an input produces wide uncertainty in every output that depends on it. The second challenge is validation. A model that includes the new bacterial variable needs to produce outputs that match observable data better than the old model did. If the updated model projects Arctic carbon absorption rates that match satellite-measured values more accurately than previous versions, that is evidence the new variable is improving the model. This validation process takes years and multiple model runs. The third challenge is the downstream revision. Every report, projection, and policy document built on the previous model versions contains numbers that may need updating. International climate reports synthesize model outputs and are used directly in negotiations about emissions targets. A significant revision to the Arctic carbon sink estimate is the kind of change that requires formal acknowledgement and correction in official scientific assessments. None of this happens quickly. Science moves through peer review, replication, validation, and consensus. The urgency is real. The pace of the process is constrained by what the scientific infrastructure can actually deliver. In the meantime, the bacteria continue working. No policy decision required, no budget approval needed. The chemistry runs in the dark, under the ice, every day. And what the next chapter of Arctic research looks like, the expeditions being planned and the questions scientists now most urgently want to answer, marks the beginning of a new phase in the understanding of polar biology. The discovery changed the priority list. Before October 2025, the central Arctic Ocean under the sea ice was one of the least sampled marine environments on the planet. Access was expensive and difficult. The assumed biological activity was low. The scientific return per expedition dollar seemed modest compared to more accessible ocean regions. After the paper, the calculus reversed. Research groups across Europe, North America, and Asia immediately began designing follow-up expeditions. The University of Copenhagen team that published the original findings secured funding for expanded sampling campaigns targeting four-season coverage, the major gap in the existing dataset. A winter expedition capable of sampling under the Arctic ice during polar night would fill the single most important hole in the current understanding of annual nitrogen fixation rates. A winter sampling campaign in the central Arctic requires an icebreaker that can operate through maximum ice thickness in complete darkness. With a science team capable of working in temperatures that drop to -40 degrees Fahrenheit on deck, the logistical requirements are demanding but achievable with the right vessel and planning. Ships like the RV Polarstern are designed for exactly this type of year-round polar operation. The ice core research question, whether nitrogen-fixing bacteria live inside the brine channel network of the ice itself, is being addressed by a separate working group. Ice core microbiology requires different drilling equipment, different sample processing, and different assay protocols than water column sampling. A coordinated expedition that samples both the water below the ice and the ice itself at the same sites would provide the most direct comparison possible. Satellite technology is contributing to the scaling effort. Ocean color sensors that measure algae productivity from orbit now provide near-daily coverage of the entire Arctic Ocean surface, including the open water areas that expand each summer. Researchers are linking the expedition's measured nitrogen fixation rates to the satellite-observed algae signals from the same locations and time periods. This relationship, if it holds consistently, would allow them to extrapolate bacterial nitrogen fixation rates across the entire Arctic Ocean surface using satellite data without needing to physically sample every location. Validating that satellite extrapolation requires more expedition data from more sites across more conditions. Each new sampling campaign feeds the extrapolation model and narrows the uncertainty range around the total annual nitrogen input estimate. Parallel work is underway on the evolutionary biology questions. Genomics teams are mining the Copenhagen expedition's genetic dataset for deeper information about the evolutionary history of the Arctic bacteria and their relationships to cold-adapted nitrogen fixers in other environments. Comparisons to Antarctic marine bacteria, deep ocean samples, and extreme environment microbes from other cold habitats are building a more complete picture of how cold-adapted nitrogen fixation evolved and spread. The modeling community is not waiting for all the data before beginning revisions. Interim model versions incorporating the confirmed findings, but with wide uncertainty ranges, are already being tested. These provisional models provide a first estimate of how much the Arctic carbon sink correction will shift global temperature projections. Those estimates are not yet published but are circulating within the climate modeling community. The ocean under the Arctic ice went from assumed desert to active biological frontier in a single paper. What the next generation of expeditions finds there will revise the global climate model. It will reshape the search for life beyond Earth, and it will tell us whether the invisible chemistry running under the ice, sustained for millions of years without human knowledge or intervention, can survive the world that is now changing above it. The bacteria do not know any of this. They run their chemistry, they convert nitrogen, they feed the algae pull carbon from the sky. And under the ice, in the dark, the cycle continues.